So we start cell organelles. As far as the different types of cell organelles have been considered, one of the very premier discussion is waiting for mitochondria. Mitochondria identified by Benda, also known as chondriosome or bioplast. Mitochondria is a very important cell organelle in eukaryotes, and hopefully enough, it is a very common term used for mitochondria, known as powerhouse of a cell. If you ask me, sir, why this name is used, as this organelle is assisting in ATP synthesis. If we go through a careful detail of the structure of the cell organelle, say mitochondria, it is like this. The whole structure is a sausage-shaped or tubular body structure, of which this part is called outer mitochondrial membrane. This seems to be the matrix. This layer is supposed to be inner mitochondrial membrane. These finger-like projections is called cristae. The cristae, whenever, is to some extent magnified like this. The finger-like projections have been supported by some tennis racket-shaped particles like this. If we magnify the structure of cristae like this, these finger-shaped long structures have been supported by these tennis racket-shaped particles, and we call them Fernandez-Moran subunit. Each Fernandez-Moran subunit is again divisible in three parts: F0, F5, and obviously F1 particle or oxysome. The whole structure of mitochondria is a very peculiar one, and most eukaryotes possess it as far as the energy generation in a cell is concerned. In certain cells, the mitochondria is again supported by another fascinating drawing. The drawing seems to be very peculiar. The mitochondria may be like this. So what is it? This is a mitochondria lacking the outer membrane, and it is called mitoplast. A very peculiar type of mitochondria possessing ATP synthetase for promotion of ATP generation. One of the very important cell organelle now I am describing is a member of GIRL. I repeat, the name is GIRL complex. According to Novikov, the GIRL complex comprises of Golgi bodies, endoplasmic reticulum, and lysosome. First of all, we go with the concept of Golgi bodies. Golgi bodies, also called as dictyosome in plants. For the first time, it was identified by Camillo Golgi. Comprises of three fundamental structural components: cisternae, vesicles, and vacuoles. Cisternae, vesicles, and vacuoles. In most of the higher eukaryotic cells of animal origin, the area where Golgi body is located, the area is definitely a very peculiar one. If you ask me, sir, why? Say this is a cell. This is the locale of Golgi body. And I must say that surrounding Golgi body, a clear hollow space you are getting, where no other cell organelles do exist, and this is known as zone of exclusion. A very interesting structure. The Golgi bodies are actually acting as a secretory component of the cell. They used to secrete a number of enzymes which act as a hydrolytic group of enzymes to simplify most of the complex biological products within a cell. So it is one of the very important cell organelle as far as the cellular secretory activities are concerned. The next member goes to ER, oblique, if I say, Endoplasmic Reticulum, identified for the first time by Pallade. The ER is supposed to be of two types. One is RER, the other is ACR. You can ask me what is the term meaning RER and obviously the term meaning ACR. RER, rough surfaced endoplasmic reticulum. And if you say ACR, obviously this is nothing but smooth surfaced endoplasmic reticulum. Now a pertinent question is this, sir, if you tell me if it is R, then a point to be noted here, the rough is rough probably due to presence of ribosomes. So endoplasmic reticulum, they are of again three types. Comprises of cisternae, tubules, and vesicles. I repeat their name: cisternae, tubules, and vesicles. If you carefully observe the cisternae of two types, cisternae of ACR and cisternae of RER, they seem to be like this. This is the cisternae of ACR without ribosome on it, and this is the cisternae of RER possessing ribosomes on it. So what makes the difference between the two types? ACR is devoid of ribosome and RER is having ribosome. But the interesting is this: RER is found in most of the highly biologically active cells where random protein synthesis is maximum. And on the other hand, ACR is supposed to be associated with these cells where there are least possibility of protein synthesis. Obviously, that has to be supported by high amount of lipid biosynthesis and cholesterol storage. Thank you very much. For testing your understanding of this lesson and more videos, log on to www.tubelessons.net.